score in this format, I need it for analysis. I'll be able to give you more insight into how to go about if I do the analysis. Okay, you have done all this. Test number, date, then what else? Questions attempted, right, wrong, positive marks, negative marks, score. All of you have done this? Okay. Now, did you all calculate your percentage score? Percentage of questions that are gone right and wrong. All of you have that number now? Those of you who have got less than 20% of the questions attempted wrong, raise your hands. Less than 20% of the attempted questions wrong. That means if you attempted 10 questions, less than 2 wrong. If you attempted 50 questions, what is 20%? 10. Less than 10 wrong. So if your questions that have gone wrong is less than 20% of the attempted questions, raise your hands. If you have attempted 50 questions, 45 is right and 5 is wrong, how do we calculate the percentage? Whatever you want to calculate the percentage of over the total number into 100, that's all. 5 divided by 50 into 100. So the number of questions wrong divided by the total number of questions attempted into 100 will give you the percentage of questions that you have attempted which have gone wrong. So do that. I want you to do some analysis first. One analysis that I want you to do for every test is percentage of questions attempted, percentage of attempted questions right, percentage of attempted questions wrong. So for this, what is the total number of questions? 180. The denominator will be 180 to calculate the percentage of attempted questions. If you attempted 50 questions, what is the percentage of questions that you attempted? 18 is 10%, 54 is 30%, 50 is somewhere between 25 to 30%. That's all. 36, 14, 140. Yeah. Around 28% of the questions you would have attempted. Correct? If it is 50 questions that you have attempted out of 180, you have attempted around 28% of the questions. So now what is the percentage of questions you attempt? Of the attempted questions, how many did you get right? How many went wrong? You need to get that. So this analysis also, this will be common. Test number, date, and this analysis. I get these things, this score I get, whatever, this data if I get, I will be able to do this analysis on Excel. We will automate that analysis so that I also get better insights. You should do it on paper. Oh, still not done. 1%, that's all. Come on. You should be doing it in your head. Okay, you tell how many questions you attempted? 75. How many did you get right? You don't remember? Total number of questions that you got right? How many questions were right? 30. 30. 45 were wrong.
Oh, sorry, 40 I got correct and 35.
40 you got correct and 35 was wrong. So the percentage of questions wrong is close to 50%. 49%. 37.5 out of 75 is 50%. 2.5 out of 75 is 1/3 of 75. 1/3 of the 10% of 75. 1/3 is 3.33. So 35 out of 75 is 3.33 less than 50. So it is 46.67% of questions you got wrong. Good? That is what we get. 46.67% of the questions is wrong. And did you attempt all the questions with some logic? What will be your score now? You attempted 75 questions. 40 is right. 160 minus 35. So 125 is your score. Alright. So 75 out of 180 is how much? If she has attempted 75 questions, what is the percentage of questions that she has attempted? 18 4s are 72. So 3 is 1/6. 1/6 is 16.25%. So it is 46.25% of the questions you attempted. Of the 46% of the questions you attempted, 46% of the questions is wrong. So the proportion, if you see, the proportion of the questions that you attempted of the total number of questions is the same as the proportion of the questions that went wrong. So that is an insight into your performance, statistical insight. Okay. All of you did that percentage. Now I want to know, any of you who have got, let us look at the range of students in terms of number of questions attempted. So again, we'll take a count. More than 150, nobody has attempted, no? 2 people have attempted more than 150. You attempted more than 150? 100 to 150. How many questions you attempted? How many? 52. 2 people told that they attempted 100 to 150. Anybody who attempted more than 150? Number of students, I need to have exact number now. Number of questions attempted more than 150. How many? Raise your hands. Nobody. 100 to 150. How many? I need to see the hands. Number of questions attempted 100 to 150. 2. Now, percentage of questions that have gone wrong. Percentage of questions wrong. Less than 20% of the questions wrong. Anybody? Raise your hands. You know how many you have attempted. You have calculated what is the percentage of questions that went right and what is the percentage that went wrong. Anybody who has got less than 20% of the attempted questions wrong? Raise your hands. Nobody. 20 to 30% of the questions wrong. 20 to 30% of the questions wrong. Okay. Raise your hands clearly. So that I can see 1, 2, 3, 4, 5, 6. 6 of you have got 20 to 30% of the questions wrong. 30 to 40% of the questions wrong. Raise your hands. 30 to 40% of the questions wrong. 1, 2, 3, 4. Raise your hand. How do I know you are just scratching your beard? What should I assume? Okay. 1, 2, 3, 4, 5, 6, 7. You're there. 8, 9, 10, 11, 12. 12 of you have got 30 to 40% of the questions wrong. 40 to 50% of the questions wrong. Raise your hands. 40 to 50%. I'll go column by column. 1, 2, 3. 1, 2, 3 up. 4, 5, 6, 7, 8, 9, 10. 10 of you have got 40 to 50% of the questions wrong. More than 50% of the questions wrong. It should be 20 then. 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. 3 people are out of statistics now. See, you have to be clear in the way you communicate, the way you raise your hands. Okay. How many is it? 18, 28. 17, no? 3 people are missing in the statistics, okay. Of those who have got more than 50% wrong, anybody who has got more than 75% wrong, is your answer. If you have got more than 75% of the questions you attempted wrong. Already. Okay. Score, final score. Final score. Anybody in the range of 400 to 600. More than 400. 300 to 400. 300 to 400. Nobody. 200 to 300. 200 to 300. Greater than 500. 400 to 500. 300 to 400. What is your score? Two? Be loud. Shout. 219. Okay. So 200 to 300 is 1. And the highest score in this test is 219. 150 to 200. How many of you have got 150 to 200? 1, 2, 3, 4. 4. 4 of you have got 150 to 200. 4 of you have got 150 to 200, 100 to 150. 100 to 150. Okay. Keep your hands up. I'll be counting by the rows, columns. Once I'm done with one column, you can put your hands down. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 of you have got 150, 100 to 150, no? 100 to 150 is 12. 17. 31 of you have got less than 100. Correct? 31 of you have got less than 100. This is the baseline study. Baseline study. Madam, have you made note of the statistics? Please make note of the statistics. This is the baseline study that we have got. I will make this 20. Whoever is missing in this statistics, for now we will make it. Once I get your marks, a better analysis will be made. This is the baseline. Clear? So the highest score in this test is 219. What is the lowest score? Any score between 0 to 10. Any score between 0 to 10. Don't worry, all of you will cross 600. We'll do a baseline study first. Alright, all of you will cross 600. Don't be ashamed of any score at this point of time. This is the baseline. Next year, when we are having the success party and we are giving a presentation in front of the Chief Minister, we have to tell the Chief Minister, no? What was your score on the first test and what is your rank in NEET? Alright. So any score between 10 to 20. Score between 10 to 20. Oh, good. 20 to 30. 30 to 40. 37. So our lowest score in this test is 36. Alright? Excellent. Very good beginning. No problem. We have got no negative score to start with. All of you are in positive score. That's a great beginning. Alright. So congratulations. Some things that I am not so happy about that you have to work on in the next test is you have to sit for the entire 3 hours. You finish attempting all the questions, read the questions repeatedly till the time is over. But you will not sit and look here and there. You will not ask the teacher to get out of the room. You will be there till the teacher says that time is up and you can move. You have to give back your paper. Until then you have to sit. Alright? And you saw the OMR sheet. Next time, before the teacher comes, you should be able to sign it clearly. Alright? Choose any random number for filling up your roll number, for filling up the question booklet number. Whatever the bubbling has to be done in terms of statistics, use random numbers for that. Every test you can use something, but you have to do that as an exercise. Your signature, name, mother's name, father's name, everything should be completed. So when the invigilator comes to you, the invigilator should have only the task of signing your OMR sheet, not telling you what to complete. Alright? Every time there will be a question, the question paper number and the answer booklet number in NEET. Those of you who have seen, the answer booklets come with the question paper. Both of that should be always same. The question paper number and the answer booklet number should be same. So in that question booklet number or the answer sheet number, there is a block there. You write some number on your question paper and write the same number on your OMR sheet. Just to get trained on looking at that. Okay, when you get your OMR sheet, you write the question paper. The first thing you will do is write some random number in the question paper, on the question paper, making that the question paper number, and the same number on your OMR sheet in the space given for question paper number or the answer booklet number. So first thing to check, before even you mark your bubbles, anything that you mark the bubbles, you will do that. Alright? Anybody who was using any other pen other than ball pen, start using ball pen, black or blue ball pen. I think the OMR sheet that we had given yesterday had a glossy paper. Glossy paper made the ink to smudge. So I'll tell the concerned teams to change the nature of paper. We'll get matte paper next time. So that issue will be sorted. But the issue should not be because of you using gel pen or pilot pen, ink pen. Use ball pen always, blue or black. Alright. Carry two pen of the same kind always. Whichever brand pen you are using, let's say you are using cello, rota pen, rota ball, whatever. The same pen should be in two number. Alright. So if one pen is not working and you are supposed to change the pen, it has to be the same pen. Another pen of the same brand. So there will be no change in the color or the shade of ink. These are the small things. Take care of that. Have your water bottles and avoid going out. Before coming to the examination hall, go to washroom, freshen up and come. Unless it is an emergency, don't go out in between. So that 3 hours, stay. Alright? You are not able to answer anything. Till the end of 3 hours, keep reading the question paper again and again. Just read the question paper. You can do that now. If you read all 180 questions once, slowly, comfortably trying to understand everything that is there in the question paper, you will need about half an hour to 40 minutes. You know nothing to answer, you can read the question paper 3 times. But no looking here and there, no sleeping, no getting your mind distracted during those 3 hours. Those 3 hours, absolute focus on the question paper. We'll do that next time. Alright? I'm happy with this. No issues. Okay. We work. Alright. Do your analysis well. Did you start doing the analysis of biology paper? Have you started making notes? Post-test analysis notes? Do that when we discuss. A lot of answers should be coming from you. So we will have those discussions. Next paper will be better. It might be possible that next paper will go down further. Because it's an initial test, no? Until you build a set of concepts around the questions that you attempt, you cannot be sure of having a sufficient base of concepts to answer questions in varying concepts, varying aspects. So next paper might not have any questions related to the concepts that you prepared on in this paper. Then that will again be a new paper for you. Nothing to fall back on your past preparation. So by the time you complete about 10 papers, or the 10, 12 papers you can consider. 12 papers, the marks can go up and down, depending on how the paper is. You make an average of your score of all the 12 papers. See, till October 31st, very crucial phase. Whatever papers, you will be attempting 15 papers till October 31st, 15 papers. October onwards, your scores will consistently start becoming better than the average score of all the 15 papers from yesterday to October 31st. Okay? That is when you have to be worried if your performance goes down. You have to start re-strategizing. Okay? You have to do more revision. Until then, keep attempting the papers, build on your post-test analysis notes sincerely. Up and down is fine. But documentation, important. We want this to keep looking at how things are moving. Clear? Oh, good. Alright. So continuing with what we were doing yesterday, laying down the foundation for understanding biology. 3 things are fundamental to all life forms. What are the 3 things? The energy and mass dynamics. The energy mass dynamics is common to all living beings. Second thing that is common to all living beings is evolution. So when we study anything, we have to look at the evolution of that organism. How has it evolved to be what it is today compared to what it was earlier. Number 3 is cell. Alright. 2 driving forces for evolution. What are the 2 forces that will drive evolution? Survival and reproduction. 3 advancements in the development of a cell, or 3 developments in the formation of a cell which accelerated the process of life formation or origin of life or the evolution of life. What are the 3 things there? Most important, if you have to say, these are the 3 things that gave life an acceleration in a cell. What are the 3 things? First one is membrane. Developing a membrane of phospholipid bilayer. Phospholipid bilayer membrane. The second one is what? That is the third one that came later. Enzymes. Cell membrane made the cell possible to regulate its activities. Development of enzymes accelerated the metabolic reactions or the chemical reactions. Number 3, formation of nucleic acids and the connection that got established between nucleic acids and protein synthesis. Alright. So everything in the body is a chemical reaction. By that, can I say me talking is also a product of chemical reactions of various kinds? Right? The lecture that I am giving you now is also a product of chemical reactions of various kinds. At various places, there may be thousands of chemical reactions happening. Alright. On the basis of the understanding that you have right now, related to metabolism and all, can you explain disorders, any disorder, take any disorder of your choice and give the cellular basis for the disorder. Disorder can be a disease or a genetic disorder or nutritional disorders, whatever it is. Tell me some disease that comes to your mind. And you have to explain that on the basis of what we have discussed so far, not going beyond. Tell me any disease, whatever disease that comes to mind right away, tell it. Whichever is the first one that I hear will be the one we will discuss. Come on, what are you thinking? You are thinking to recall some disease that you got? Asthma. Okay. Explain it in a cellular level, at the cellular level. What is asthma? What is asthma? To come in. stand turning to the wall. Don't go there. Stand, I mean, turn towards the wall. Turn towards the wall. Normal response. Okay, turn. You do that now to me. You do that now to me. That is the immune system. Normal response is not getting hyper about some stimuli. So the stimuli, I called it. Normal response is to look at who that is and then decide what to do. Immune system getting hyper to some stimuli is an allergic response. A reaction, a stimulus to which there is no need to overact. When some overaction happens, that is allergy. Asthma is an allergic response. Asthma is an allergic disorder where the lung tissue is responding in a, the lung tissue, the immune system in the lungs is responding with a hyper reaction. It's a hypersensitivity disorder. That's all. Some people, most of the people are comfortable around nature. Some people, as they go into go amidst plants or in a field during pollen season, they will start sneezing, rhinitis, allergy, a running nose. They will start. There will be redness in the eyes. Some people will have allergy to some oil, allergy to parthenium. Right? You eat some oil. Some people have peanut allergy, groundnut allergy. They eat peanuts or anything with peanuts, they will start developing reactions in the body. Allergy to egg. These are all common things. You eat egg every day, they are keeping egg every day. So you're eating eggs every day, there is nothing happening. Some people might die because of eggs. Because the immune system is responding in a hyper-reactive manner. What is immune system made of? What is immune system made of? Immune cells again. They are also cells only, no? So the response is production of something. See, the immune system is made up of cells, the white blood cells, WBCs. The cells will have certain receptors on their surface, like what somebody told when we were talking about enzyme reaction. What will the enzyme have for the substrate? Active site. The enzyme has an active site. The immune system is also trained to identify certain things. It is trained. Like I told you, nothing happens just like that. Nothing happens by accident, by chance. No, everything has got connection. So immune system is supposed to identify something and react to it in a pre-decided manner. Pre-decided manner based on evolutionary training that our immune system has got. Life is in continuity, no? We have been living for the past 3.5 billion years. All of us have been living for the past 3.5 billion years. So all of you might be in the range of ages 18 to 20, 21 maximum. I am 40. So by age, I am double your age. But in terms of life, all of us are of the same age. The life in us has been existing for 3.5 billion years now. Since the time life originated, life gives rise to next life. Gives rise to next life. So life is being passed on continuously. Yes, sir. Before you were born, whatever genetic material you have, the life you have was in the sperm and egg of your parents. When those two cells, those two are living cells, two living cells came together and formed one living cell. And that living cell became you. Where were you? You were existing. No? The life in you existed even before you formed as the zygote. It was existing in the cells of in the gametes of your parents. Where were they? They were also existing. So if you notice, the genetic material is being passed on in continuity. Since when? Since it formed. So the genetic material is a series of codes. Chemicals basically, adenine, guanine, cytosine, thymine, or uracil if it is RNA. But what gets passed on is DNA. Now, adenine, thymine, cytosine, uracil, 4 alphabets. being repeated in a continuous sequence in various ways. A copy of that code is taken from the parent, the father, a copy of that is taken from the mother, they both come together and you have a copy of your own alphabets. But is there anything new in that? It is a recombination of what your parents had. That means, if we consider the DNA that you have in your cell as the library, it is a library which has information of everything that has happened for the last 3.5 billion years. In the line of exposure that your ancestors dating back to 3.5 billion years have got. Correct? You are an evolutionary library. So if anybody can understand what the DNA, one set of DNA in one human being's one cell means completely, they understand life in its entirety since the beginning of life. Are you understanding the scale of it? So where is all this? It's all in the cell. It's all in the cell. How you react, how your cells react, all of that is already designed within the cell. And it's all happening in terms of chemical play. What gets produced? Chemicals. Who produces it? On what basis does it get produced? On what basis does do the receptors on the cell gets produced? Where is the basis for that? Where is the blueprint for that? The blueprint is in the DNA. RNA will read the blueprint, translate that into proteins. Is that the protein which will go on the surface of the cell? Maybe, may not be. Most of the proteins that are produced by the translation of genetic code are enzymes. You understand? Most of the proteins produced are enzymes. It is like if all the chemical play that is happening inside your body is considered to be a big restaurant, a massive restaurant feeding millions of people, the DNA or the new DNA is like the MasterChef Academy. It is not the restaurant. DNA is not the restaurant. But sometimes whatever is cooked in the academy is also served, no? What will happen to the food that is cooked in the academy? Will it get thrown? It can also be served. But is the purpose of the academy to produce food to be served to millions of people? What is the purpose of academy? The purpose of the MasterChef Academy is to train the chefs who will go out and work in different restaurants in this whole city of restaurants which feeds millions of people. Some restaurants might serve one particular cuisine, some restaurants might serve something else, something might serve something else. Based on where the restaurant is and what it is supposed to cater to, ingredients will be used and those ingredients will be mixed and matched to produce the dishes which will be served to people who are supposed to eat it. You understand this restaurant? That's all, no? But are all the chefs capable of, no, not the chefs, anyways. Can you correlate now? The MasterChef Academy is what? In your cell? The MasterChef Academy is? The MasterChef Academy is? Nucleus or the DNA. You can consider DNA because we will remove the boundary. We will consider it as DNA. Nucleus is just a boundary within which the DNA is present. DNA is the MasterChef Academy. Enzymes are? What are enzymes? Enzymes are? What are the enzymes? Enzymes are the chefs who are produced in this academy. The dishes are your entire body. Whether it is hair, nails, enzymes, the digestive enzymes, all of those are the dishes. Alright? That's all. Go one step further. Can I say what my body can do or what I can do or cannot do is dependent on my enzymatic machinery. Will I be right or wrong? The limit of the ability for this body and mind also is the enzymatic machinery that I inherit. Yes or no? Why do we have differences? Why some people grow tall, why some people are short? It is the enzymatic machinery that will decide what kind of proteins can be produced, what kind of products can be produced, how the body can be organized. Yes or no? Color of the eyes. If you have the enzymatic machinery to produce blue color pigmentation, then your eye, the eyes will be blue and your eyes will look blue. Enzymatic machinery to produce brown color pigmentation, it will be brown. So if our body is an ocean of chemicals, what are the chemicals which will create meaning to the ocean to give it a form? What are the chemicals that is contributing to the creation of form and function? Which chemicals are giving contribution in this? Enzymes. Correct? Are you understanding, everybody? Yes or no? Yes or no? It should be either yes or no. Yes? Alright. I understand you are all very calm people, not very expressive. But I have failed as an actor in front of camera. But I have done well in theatre, on stage. So a theatre artist always looks for cues in the audience. So give some cues. I don't want anybody to not understand. I want everybody to understand what is going on so that you are on the same page of our discussion. But if I don't know whether you are understanding or not, problem. And I don't want to assume. Okay, we'll set some basic operating principle so that I don't force you to change your nature of being calm and become aggressive. If you don't understand anything, whether in my class or any of the classes for the entire duration of the program, you will raise your hands. Raise your hands, not like this. Then come and tell, sir, I wanted to ask you a question, you will not give me a choice. This I will not understand as raising hands. Raise your hands and you will ask it. Alright? Any time of the discussion you will ask. You will not let the discussion move forward if you have not understood. Is that okay? Alright. Done. So cell, what did we understand now? What is this? With the cell, so many terms come related to the cell. When we are discussing molecular biology, when we are discussing genetics, all of that is also related to the cell only. We are discussing some functions that are happening inside the cell. So many terms come. Like I'll give you some terms. Let me see if you can give the difference. I always prefer to understand the story and then fill in the details, rather than trying to go in a hurry to know the details of the story. Right? Get the story. Forget about the names. Get the context of the story. Then you can add on the names. If you focus on remembering names, terminologies, definitions and all that, it will become difficult for you to get the story. You don't need to know any terminology to understand the story. Okay, it will become a layman story. Okay. I'll tell you a story. Let's see. I'll tell you a story. You have to tell me what does it relate to. Okay. There's an estate in a big country. Estate in a big country has various kinds of people working in this estate. This estate has a set of people who have some specialized training. And these are all very happy people, focused. They wake up early in the morning, they go through their training, they do their job and all that is happening. 2 newborn babies are born into this family of these workers, these disciplined workers. The babies grow, they grow faster. They grow and they are looking around, they are playing around. Suddenly in their part of the estate, there is an alarm that beeps. there is an alarm that goes off, and there is an announcement made calling for a few of their members. They are identified, and the elders wish them. They cross a particular barrier and they go. The kids want to see what is happening, and they just go in, and they are blocked. They are not allowed to go. They come back. They think something is happening. Few days pass by, and these people don't return. And the kids are told that for the welfare of the estate required food. The estate required food for taking care of all the people in the estate. So these people had to sacrifice themselves to get food. To make food available, these people had to sacrifice themselves. Why the kids are surprised? Why do they have to die to make food available to the people in the estate? Now okay, time passed by, they forget that. This happens again. This happens again, and alarm sounds, and there is a message given that there has been a chemical attack on the estate, and some of these people have to go and fight it. These people go, they fight it, and in the process, they also die. Some of them die again. Kids are surprised. Now they are starting to realize that anybody who crosses this gate and goes never comes back. Elders in the community know this, but these kids are learning this. But they are also curious to know what is going on. Third time an alarm beeps. Now the kids are a bit worried. They are anxious. Who will be called now? They are assuming that these people also, when they go, they will not come back. And the elders announce that there has been a lot of garbage that is there in the estate, and this garbage has to be cleared. So some of our people have to go. But the thing is, they will also die. Then there is a different kind of alarm that will start beeping, and all the kids, everybody of this community are called for a meeting by the elders. Now the eldest of the community, the leader of this community, starts making a speech in a very bass voice. My dear brothers and sisters, all the kids, now the time has come to kill our master. So far we have dedicated our lives in the service of our master, but our master is becoming harmful for the nation. So the time has come now to kill our master and destroy this estate. In this process, we have to give up our lives. All of them say, "Jo bole so jaha." You understand? End of story. Who is this? Who does this story relate to?
So, I have two theories. One is the first two, I think you are talking about white blood cells.
Okay.
And the third one, I got lost when you said that they wanted to kill the master. I got lost there.
You got it?
Yeah, I got the first two, but the third one I don't know.
In fact, the last one is what should give you the answer. Last one is what should give you the answer. This community took care of 4 things. What are the 4 things that this community did in the first instance? Why did they give themselves up? For what? To make sure food is made available to the estate. Second, what happened in the second instance? There was an attack. So they were supposed to take care of removing those dangerous chemicals. Third, what did they do? Garbage clearance. They were supposed to clean the estate. Fourth one, they destroyed the estate only. Who is this? What is the self? In all this story, there is estate and there is a community. Community which is doing a lot of work, and they do so many things. Yes. Huh? What is the lysosome? Estate or the community? Community is lysozyme. Lysozyme or lysosome? Lysozyme is the enzyme in the eyes. It is the tear enzyme. That is the antibacterial enzyme in the tears. Lysosome. Does that fit into the story? Then what is the estate? Estate is cell. Tell me the functions of lysosomes now. Lysosomes are accused of being the suicide bags of the cell. They should be given Bharat Ratna of the cell for being the protectors of the cell. First thing, how do they make food available? How do they make food available to the cell? Lysosomes participate in phagocytosis. When the cell ingests some food material into the cell by phagocytosis, that cannot be utilized by the cell just like that. It has to be digested first. So lysosome contributes for intracellular digestion. Second, if there are any unwanted substances that enters the cell, lysosomes are the ones which will go and fuse with them, destroy that, make a destructive vacuole, merge with the cell membrane and pushes it out along with all its own content. So it is gone. Number 3, the accumulation of cell metabolic debris. Cells will accumulate some metabolic wastes now. As a lot of metabolism is going on, lysosomes have the role of clearing the metabolic wastes also. And 4, apoptosis. Apoptosis is programmed cell death. Programmed cell death meaning when the time comes that the cell can no longer be repaired, the cell has been broken down beyond repair. And the cell, if it survives, will become harmful to the body. Body is the country. The cell becomes harmful to the body, then lysosomes are the ones which will go and kill the cell. One more, 4 functions. You can tell this story, you can enact a play on this story without even talking about cell or lysosome or the cellular functions. Even 5-year-old, 6-year-old kids will be engrossed in the story, they will enjoy the story. All that you have to do after telling the story is correlate. And you remember, no, that's all. So, the way I take you through the subject is, we need to get the story first. You need to understand how things are moving, how things are functioning. Then we will work on building the specific details of the subject, which will help you in identifying terminologies. It all comes down to terminologies only, that's all. You know the story that the cell has something in itself which can actually kill the cell if the cell is useless. That you can remember. That something is a term. Lysosome is something you have to remember. There you have to put in some repetitive effort to remember the name. Otherwise the story is fine. If you understand, you will not forget the story. The names, if you don't revise, you will forget. Okay? Okay. Another example I will give you within the cell. Tell me the difference between DNA, okay, we will do this later. I will write the entire sequence. DNA into chromosome. DNA, chromatid, chromosomes, sister chromosomes, homologous chromosomes. Difference. Okay, let's do it now. DNA, chromatid, chromosome, sister chromosome, homologous chromosomes. Can anybody tell me the differences? Today when we will be discussing cell biology, I have planned to, if time permits, discuss about cell cycle also. Cell cycle and cell division. To understand cell cycle and how cell divides, these terms will come in that chapter. DNA, chromatin, chromatin chromosome, chromatid, sister chromosomes, homologous chromosomes. You need to know the difference between these structurally. What is the difference? How much time you would have spent in memorizing these things? Right? DNA is genetic. Okay, then. What is chromatin? Chromatin is sterilized structure of DNA. Huh? You get this right.
Thread-like structures in the
Spell it.
Thread-like.
Thread-like structure of DNA. Okay. What is chromosome?
Condensed form.
Very good. Condensed form of DNA. Okay. Okay. What is sister chromosome? When two daughter chromosomes. Okay. Obviously, both the daughters are sisters to each other. Okay. What are homologous chromosomes? You are right. See, take a thread. Take one thread. That is the DNA. You take small balls and coil the thread around these balls. How many coils? How many coils? 1.5 coil. You coil the thread around these balls. Now the same thread which is coiled around these balls is chromatin. That's all. What are those balls? What are those balls made of? Proteins. What proteins? Histone proteins. How many varieties are there? 1, 2A, 2B, 3, 4. What is this? How many proteins are there in this ball? 8. Okay, it's a histone octamer. We can fill it up later because you already have studied. I am telling that. Otherwise, it's just simple. You have to teach this. You have to teach these things to a small kid or a 5th standard, 6th standard kid. Take a yarn. That yarn is there, no? The woolen yarn. That is DNA. Coil it around balls. That is chromatin. Fold it. Fold it and condense it. By folding it, that is chromosome. That is chromosome. Take a photocopy of this. Exact. When I say photocopy, exact copy of this, you are making a copy of the same, duplicated. When they are together, they are still a chromosome. And each of the parts of the copy are chromatids. And if the chromatids are of the same chromosome, then they are sister chromosomes. You pull the chromatids of the chromosome apart. Now what do I have? I have 2 chromosomes. I bring these 2 and attach them. What do I have? How many chromosomes do I have here? 1 chromosome. What do these 2 become now? Chromatids. And because they are of the same chromosome, they are sister chromatids. I pull them away. What are they now? What do I have? Do I have 2 chromatids? No. What do I have? I have 2 chromosomes. How many chromatids are there now? How many chromatids are there? 0. I have 2 chromosomes. Alright. Now let us consider this index finger of mine is the DNA. Now I coil this with protein. What do I have now? Coil this around protein. What do I have? Chromatin. Now I have condensed it. Let's say this is the condensed form now. Imagine this index finger to be 2 kilometers long. Now I have condensed it and make it this much. This is chromosome. But before I condensed it, I had made a copy of it also. I had made a copy of this. Now as I condense, the copies become visible. Now, what do I have? Chromosome and chromatid. What do I have here? Condensation of the nucleus and appearance of chromatids. For every nucleus, every chromosome, we see the condensation of the chromosome. Chromosome condenses and chromatids start appearing. Metaphase, lining up of the chromosomes. Lining up of the chromosomes along the equator. Anaphase, separation of the chromosomes. That's all now. Over. I want you to tell me in terms of the amount of DNA. This is 100 units of DNA. So both the overall, let us assume the overall DNA content in this cell is 100 units. So at this stage, how many units of DNA do we have? 100 units at the beginning. By prophase, here how many units of DNA do we have? 200 units. See, don't consider this as 100 and this as 100. Together this cell has 100 units DNA. Now at this stage, how many units of DNA is there? 200. Here how many units of DNA is there? 200. Here, how many units of DNA is there here? 200 only. Now here, 100 plus 100. Both the cells got 100 plus 100. So this is the parent cell. These are the daughter cells. Right? Now look at this here. How many units of DNA in meiosis 1? And okay, one more thing we will read. How many chromosomes are there here? How many chromosomes are there at this stage? How many chromosomes are there here? 0. Why? Only when the chromosomes are clearly visible in condensed form, when the DNA is visible in the condensed form, you call it chromosome. Correct? How many chromosomes are there here? 2 chromosomes. How many chromatids are there? How many chromatids are there? 0. How many chromosomes are there here? 2 chromosomes. How many chromatids are there? How many chromatids are there? 4 chromatids. Do you see this? You have 4 chromatids. That is, you have 2 pairs of sister chromatids. Then here, how many chromosomes are there? 2 chromosomes. How many chromatids? 4 chromatids. Here, how many chromosomes? 2 chromosomes. How many chromatids? 0. Here, in the other cell also, 2 chromosomes, 0 chromatids. So do we have, and when we decondense it, it will again become 100 units of DNA, no chromosomes. Mitosis is not just maintaining the number of chromosomes, it is maintaining the quantity of DNA, the amount of DNA that is there in the cell. That is important. The amount of DNA in the parent cell and the daughter cells should be the same. We look at meiosis. Meiosis 1. How many units of DNA is there here at the beginning of it? 100 units of DNA. How many chromosomes? 0. Here, how many units of DNA? 200 units of DNA. How many chromosomes? How many chromosomes? 2 chromosomes. How many chromatids? 0 chromatids. Here, 200 units of DNA. Same so far, till metaphase. It will be the same. 200 units of DNA. How many chromosomes? 2 chromosomes. How many chromatids? 4 chromatids. Here, how many units of DNA? 200 units of DNA. How many chromosomes? 2 chromosomes. How many chromatids? 4. How many units of DNA? 100 units of DNA. How many chromosomes? 1 chromosome. How many chromatid? 2 chromatids. 2 chromatids. Here, 100 units of DNA, 1 chromosome, 2 chromatids. Now, what has been reduced in meiosis 1? There are 3 aspects we are looking at. The amount of DNA, the number of chromosomes. Amount of DNA, has it been reduced compared to the parent cell? No. At the end of meiosis 1, the number of chromosomes that the cell can condense has been reduced. But the amount of DNA is still there. It is still the same. Now this cell becomes the parent cell for meiosis 2. This cell becomes the parent cell for meiosis 2. I will not draw it. I will just write the amount of DNA and chromosomes. Is it okay? In meiosis 2, at the beginning, do we have interphase? No interphase. How many amounts, what is the amount of DNA that we will have at the beginning? 100 units of DNA. How many chromosomes do we have? 1. How many chromatids? 0 yet. It would have decondensed. Now it has to still appear. Here, 100 units of DNA, 1 chromosome, 2 chromatids by the end of prophase 2. In metaphase 2, how many units of DNA? 100 units of DNA. How many chromosomes? 1 chromosome. How many chromatids? 2 chromatids. Here, what will happen now? 50 units of DNA, 1 chromosome, 0 chromatid. 50 units of DNA, 1 chromosome, 0 chromatid. Now, look at the 2 things. Has the amount of DNA reduced in meiosis 2 compared to the parent cell? Between the parent cell and the daughter cells, parent cell has 100 units of DNA, daughter cells are having 50 units of DNA. Has the number of chromosomes reduced? No. Number of chromosome in the daughter cells is the same as what it was in the parent cell. How is it like mitosis? How is it like mitosis? Meiosis 2 is also reductional. Alright. In mitosis, there is no reduction in terms of either the amount of DNA or the number of chromosomes between the parent cell and the daughter cell. So in this process there is duplication, separation. So the amount of DNA as well as the number of chromosomes remain the same. But in meiosis 1, what is reducing? There should be 2 types of reduction that needs to happen. Number of chromosomes and the amount of DNA. Which one is happening in which phase is what we have to be clear. In phase 1, in meiosis 1, what is being reduced? The number of chromosomes are reduced. In meiosis 2, what is reduced? In meiosis 2, what is reduced? The amount of DNA. So at the end of it, at the end of both the phases of meiosis, the daughter cells should have half the amount of DNA and half the number of chromosomes. It is not just half the number of chromosomes, it is half the number of representative chromosomes. What do you mean by representative chromosomes? We are what kind of organisms? We are very loud. I told you, you should have some kindness towards physically disabled people.
Male and female.
Huh?
Male and
Not male and female. We have 2 sets of chromosomes. We have 2 sets of chromosomes. What do you call that? Diploid. We are diploid organisms. Okay? Diploid organisms have 2 sets of chromosomes. So in meiosis, the daughter cells should have 1 set of chromosomes. It is not just half the number of chromosomes. When I say representative chromosome, ploidy division should happen. It is not just the number of chromosome division. It is not just the reduction in the number of chromosomes. It is the reduction in the ploidy. Diploid cells goes through meiosis and become haploid cells. So in diploidy and haploidy, it is not just the number of chromosomes being half, it is the number of sets of chromosomes being reduced to half. So haploidy duplicating is diploidy. Two haploid cells come together, that is why it becomes diploid now. That is why in sexual reproduction, one set of chromosomes, one set of genetic material from the father, another set of genetic material from the mother comes together, ploidy is restored. When ploidy is restored, growth begins. Alright, reduction is happening to ensure restoration of ploidy when there is sexual reproduction. This is the basis for reproduction, cell division, genetics, molecular biology. So if we, how to tabulate this? This will go. Tabulation needs to be done on this. Mitosis, meiosis and meiosis 2. Meiosis 1 and meiosis 2. The same representation can happen in the form of a table. You will write all the phases and you will write 3 columns for each. Amount of DNA. For the purpose of understanding, we will say the beginning state is 100 units. Whatever is the unit, 100 units is the beginning state. Then the number of chromosomes, number of chromatids. For every stage, you will represent it that way. So you will have the first column having the stages, the second column, and the 3 columns for each of the cell division type. Mitosis will have 3 columns: amount of DNA, then number of chromosomes, number of chromatids. Alright, that's all. We'll do that. I'll just write the table, you have to tell me the numbers. Phase, mitosis, meiosis 1, meiosis 2. DNA, chromosomes, chromatid, DNA, chromosomes, chromatid, DNA, chromosome, chromatid. All right. Interphase. Interphase. What is the amount of DNA in mitosis? Come on, tell me the numbers now. 100 units. Chromosomes. 0. Chromatid. 0. Meiosis 1. Interphase. DNA. 100. Chromosomes. 0. Chromatid. See, now there is another issue here. While tabulating this, let us make an assumption that we are doing the referencing for end stage. If the parent cell had 100 units of DNA, by the end of interphase, how much will be the amount of DNA? 200, no? DNA duplication is happening in which phase? DNA duplication is happening in interphase, not in the prophase. So, we will make the assumption here. Parent cell For any table you have to make the assumption. At the top of the table you write assumption. Parent cell has 100 units of DNA. Parent cell has 100 units of DNA. Parent cell has 100 units of DNA, and the phases that we are mentioning now is the end of that phase. So whatever we have to write, we will be writing it at the end of the phase. Now tell me, at the end of interphase, if parent cell had 100 units of DNA, what will be the amount of DNA? What will be the amount of DNA? 200. Because DNA has become double. In interphase there is DNA duplication, DNA replication happening, DNA has become double. Okay. DNA has become double. Is that clear? What is the DNA content at the end of meiosis, interphase of meiosis 1? 200 again there. Chromatids 0. At the end of meiosis 2, at the end of interphase, what is the amount of DNA? There is no interphase. There is no interphase. There is no interphase. Correct? Now, at the end of prophase, at the end of prophase, at the end of prophase, what is the amount of DNA in mitosis? Same, 200 chromosomes. Now again, one more assumption is 46 chromosomes, 23 pairs. We are considering human beings. Mitosis and meiosis in human beings. Alright. So at the end of prophase, the number of chromosomes, mitosis, 46. Number of chromatids. Number of chromatids. At the end of prophase, number of chromatids. By the end of prophase, chromosomes are clearly condensed and the chromatids are visible. So how many chromatids will be there? So at the end of prophase, how many chromatids are there? If there are 46 chromosomes, 92. Each chromosome is having 2 chromatids. Understanding? Here in meiosis 1, or we'll do that in the continuity, meiosis 1 and 2. We'll finish mitosis. Metaphase. Amount of DNA, 200 chromosomes at the end of metaphase. 46 chromatids, 92. The only difference is chromosomes have lined up at the equator, along the equator. Anaphase. DNA
100 plus 100.
100 plus 100. Chromosomes. Chromosomes. 46 plus 46. Chromatids. Chromatids. 0. So you separated the chromatids. They became chromosomes. Now they are no longer as, you don't find chromatids. At the end of telophase, what do you have? 2 cells with, we'll change that. Hello? Yeah. At the end of telophase, what do we have? 2 cells with DNA, 100 plus 100 chromosomes. At the end of telophase, 0, chromatid 0. End of telophase is like the beginning of prophase. Correct or reverse? Chromosomes will decondense, nuclear membrane will form, and the chromosome will become diffuse chromatin network. That's all. Clear, everybody? Now we'll do this, do it for this. Prophase, at the end of prophase, meiosis 1, DNA, 200, chromosomes, 46, chromatids, 92. At the end of metaphase, DNA, 200, chromosomes, 46, chromatids, 92. Going same so far. At the end of telophase, DNA, 100 plus 100 chromosomes, 23 plus 23 chromatids, 46 plus 46. Each of these chromosomes are still, each of these chromosomes have their chromatids yet. The twins have not been separated. The couple are separated now. So the chromatids are 46 plus 46. So at the end of telophase, amount of DNA 100 plus 100, number of chromosomes 0, number of chromatids 0. When I say chromosome, physically it is appearing or not, that's all. We don't see it yet. Alright, the reduction has happened in the number of chromosomes. The reduction has not happened in the amount of DNA. Clear? No interphase in meiosis 2. At the beginning of prophase, at the end of prophase, what is the amount of DNA? Meiosis 2? 100. We are looking at it for every cell, every daughter cell of meiosis 1. Chromosomes? 23. Because each of these daughter cells here at the end of meiosis 1 has 23 chromosomes. Chromatids? 46. The metaphase, 100, 23, 46. At the end of anaphase, 50 plus 50, 23 plus 23, 0. At the end of telophase, each one will have 50 plus 50 units of DNA, no chromosomes visible, no chromatids visible. Clear? The same thing, what we discussed previously, we are representing it in terms of numbers. To show these numbers to somebody without explaining to them what story we had before this will not make sense much. Alright, now you understand this? Is it clear? Two assumptions we have made. One assumption is the parent cell has 100 units of DNA and it is a human cell with 46 chromosomes deployed. That's all. Clear?
Can you explain again anaphase of meiosis 2?
Anaphase of meiosis 2.
Number of chromosomes. Number of chromosomes.
At the end of meiosis 1, we had two cells with half the number of chromosomes. Correct? The amount of DNA was same. Human cell is supposed to have 46 chromosomes. The daughter cell at the end of meiosis 1 has 23 chromosomes. Why did we not stop here? Why should there be meiosis 2 also? Why should there be meiosis 2? We got half the number of chromosomes. Why did we not stop here? Why should there be meiosis 2? Why? Because the amount of DNA is the same as that of parent cell. So if this cell becomes the gamete, then the resulting zygote will have double the amount of DNA. if this is the end of meiosis, reduction and division, every generation will start having double the amount of DNA as the previous generation, which will not continue to be the same species. So, chromosomes have been reduced, but we have to reduce the amount of DNA. That is why meiosis 2, there is no interphase. Why? In interphase, what will happen? Doubling of DNA will happen. We don't want that to happen. We want to reduce whatever is there. We don't want any doubling to happen. So meiosis 2 does not have any interphase. Starts with prophase. What will happen in prophase? The nuclear membrane dissolves. The chromatin will start condensing into chromosomes. When the chromosomes condense, here we separated the chromosomes. We did not pull away the chromatids. So every chromosome has 2 chromatids already in the opening stage, in the beginning stage itself. When it comes to meiosis 2, every chromosome is still having those 2 copies. Duplicates are still there. The twins of the chromosomes are still there. So in prophase, when we condense, chromosomes condense. How many chromosomes are there? 23 chromosomes. Each chromosome is having a pair of sister chromatids. So the total number of chromatids we have here is 46. Metaphase, how will they line up? Will all the 23 chromosomes line up in a single line or double lines? Single line. So lining up is similar to mitosis. The lining up is of all the 23 chromosomes. When they are lining up, in anaphase, what do you do? The chromatin fibers, not the chromatin fibers, what fibers? Centromere fibers, middle fibers, pull away the sister chromatids. As soon as they get separated, what are they? They are chromosomes. So we have 23 chromosomes lining up in the center of the equator during metaphase, at the end of metaphase. Ropes are being put, spindle fibers are put on both the sister chromatids of every chromosome. You pull the chromosomes on from either side, the sister chromatids separate. You have 23 of those coming on this side, 23 of those coming on this side. When they detach, they no longer are chromatids. Okay. Now, what is the mechanism that they have adapted, those plants? See, all of this should happen at the same time. During photosynthesis, stomata should open, carbon dioxide should enter during the daytime. During the daytime, light-dependent reaction is happening, energy is being harnessed. Parallelly, carbon dioxide should be let into the leaves. The Calvin cycle should happen. This energy is used, Calvin cycle is worked on, and photosynthesis happens. All of this should happen during the daytime. Though Calvin cycle is light independent, but it can work during daytime only because it is coupled to the light reaction. It is linked to the light reaction. What do you do? Die without water or die without food? The adaptation is crossylation acid metabolism, CAM photosynthesis. These plants, they will open their stomata during the night time. So carbon dioxide goes inside the leaves. They will use that carbon dioxide and fix it into different carbon-based compounds. What is the compound? Do you remember? Malic acid? Malate? No? What is the compound? Huh? Oxalate? Let it be. So it will fix it, if the carbon dioxide is fixed into a carbon-based acidic compound. Crossylation acid is a cluster of compounds. A variety of compounds are called crossylation acids. Now, fix all the carbon dioxide that you can fix during the night time. Close the stomata before sunrise. And during the daytime, you don't have to open stomata for light reaction. Light reaction will happen, and this carbon dioxide which has been fixed into this carbon-based compound, through a series of steps, will be released internally within the leaves. So carbon dioxide as a gaseous compound is made available for the dark reaction or the Calvin cycle. See the adaptation. Nature is so beautiful. It makes all kinds of adaptations. Okay, so it was not supposed to be going. I was not supposed to be going into so much detail. Photosynthesis and respiration we will elaborate and understand in context. It is reversal. You reverse photosynthesis, that is respiration to an extent. Both of those have electron transport chain. And for electron transport chain, we have to understand redox reactions, reduction oxidation reaction. Reduction oxidation reaction both in terms of movement of electrons from high energy state to low energy state and also transfer of protons. If you transfer proton, are you reducing something or are you oxidizing something? So if I add H plus to something, am I reducing it or oxidizing it? Reducing it. When I remove H plus from something, it is oxidizing. So if I am removing H plus proton from something, I have to give it to something now. Simultaneously, something is getting reduced, something is getting oxidized. A series of these reactions is the electron transport chain, both in photosynthesis as well as respiration. When we do those two chapters, one session will do that. That will give you the basis for understanding the entire energy dynamics. Then we will have to do evolution and origin of life in detail and correlate evolution in all the topics that we study. Then cell biology. Once we are done with these three in detail, wherever we go, discussing the questions, question-based discussions, all of that will add on more and more knowledge to your understanding. Okay. These things we will do it. I will do it in an elaborated manner. So we will have that. Okay. Okay. We will touch upon a little bit of biomolecules, cell biology, biomolecule basis also we need to understand. Okay. Is it okay to sit through the entire 3 hours or we need to schedule a 10-minute break in between? Though it's not a continuous 3-hour lecture, there is a lot of discussion breaks also that is happening. Right. I am an old school trainer who believes that you have to train your body first. Train the body to listen to your mind. So, no pain, no gain. Those things work on my mind. And get used to the pain. All those things that you will find in the gym, no? It doesn't get any easier. You get stronger. All those things. Okay. For a few weeks we will have, it's okay, no problem. If anybody wants a break in between, take it. Otherwise, sit. Why 3 hours, 3 hours, 3 hours? To sit at one place for 3 hours is the first practice that we have to do. Example. So you will get to do that twice in a day. One session in the afternoon, and again you see, 9:30 to 12:30 is the first 3-hour session. 5:30 to 8:30 is the second 3-hour session. Technically it should have been 2:30 to 5:30 or 2 to 5. But 2 to 4 I have kept, and I don't want that session to be the 3-hour session during practice, because more likely you will sleep off if it is for 3 hours post lunch. Okay. This is the bit, no? Sounds like thunderstorm. Okay. See, let us understand biomolecules in the context of our survival. Okay. There are 5 classes of nutrients that we require to survive. What are the 5 classes of nutrients that we require to survive? Carbohydrates, proteins, lipids, vitamins, minerals. Simple. Carbohydrates, proteins, lipids, vitamins and minerals. Where are we getting all of this from? The source of entire mass of this body, can I say, is the earth. Material, the physical material that we have in our body is all coming from the earth. Carbon is coming from predominantly atmosphere. Correct? Some carbon based compounds are taken from the earth also. So plants are taking in the material from the earth, using sunlight to produce energy. They have the mechanism to do manipulation of the elements and create matter. We also create matter. We also utilize energy by breaking down food and releasing energy by breaking the bonds. We also get material from the food that we eat. Our body has the intelligence, genetic intelligence, to use that material and design matter that suits the design of this body. Correct? If I eat bread, that bread will contribute to building this human body only. If the same bread is eaten by a dog, the same bread will contribute to building the dog's body. So what is building is the intelligence that is there inside the individual body. And where is that intelligence? Where is that intelligence? In the genetic material. How did that intelligence come? Through millions of years of evolution. Through millions of years of evolution. What is the requirement of survival for this body? Survival requirement we have to understand. That will tell us why these are the biomolecules and why they are important. What all is required for this body to survive? What are the requirements of this physical existence? First, growth. Correct? All of us were 1.5 to 2 kg babies. Now, we have eaten so many babies. Alright. Growth, that means we have to accumulate matter, accumulate mass, and that matter has to be converted into mass within this body. For that we need to understand what is it made of. What is this body made of? Correct? Skeleton has minerals. Skeleton has a lot of minerals which form the framework of the skeleton. Minerals integrated into the skeleton. The muscular system has proteins. All our machinery that is required for carrying out all metabolic processes, enzymes, are proteins. Enzymes are proteins. We have to build our nucleic acids. So if we go on doing the elemental analysis, if we break down all this body is made of, if we look at it in terms of elements, what are the predominant elements that we find in this body or all forms of life on the planet? What elements are commonly found in all life forms? Carbon, hydrogen, oxygen, nitrogen. Correct? We need to get these 4 elements. Carbon, hydrogen, oxygen, nitrogen. These are the 4 elements we need. Proteins are nitrogenous. So no, nitrogen will help in building the proteins. We have a variety of minerals that is required in different places. Our bone requires what mineral? Bone requires calcium. Blood requires heart. The pumping of the heart is dependent on the availability of one mineral. What is that? Potassium. Nervous system cannot function if there is no sodium. Nervous system will break down if there is no sodium. There are sodium channels, potassium channels, calcium channels. Muscles need calcium to function. Correct? Molybdenum is required for blood cell formation. Copper is required for some other function. So we require a variety of minerals in various places in small quantities. So those minerals are necessary to carry on the functioning of this body properly. What do we need vitamins for? What do we need vitamins for? So we know this body is carrying out a lot of chemical reactions. All the chemical reactions are mediated by enzymes. These enzymes require energy for carrying out the reactions. So we need energy. Energy is one requirement. Energy will come by breaking down bonds. So we need to eat something that the body is capable of breaking down and taking energy. What is that? That which we have developed over millions of years of evolution. What to eat is evolution. Alright. So it is always better to eat what your family has been eating for generations than to go on changing your dietary patterns. What your family has been eating for generations is the best food for you. You can taste everything, but you should not change your food habits completely. So you go to America, you should not start eating all those, cheese-rich breads and all that. You should somehow find out a way to eat what your family has been eating forever. That will keep you healthy. Alright, clear? So we need energy. The entire machinery is dependent on proteins. We need to supply the raw materials necessary for building proteins. Our body is a thermally insulated body. Thermal insulation is there in the body. What is this thermal insulation made of? Fats. Thermal insulation. Neurons have fat around them. So conduction, nerve conduction, electrical conduction, communication in this system requires cushioning and insulation, fats. So energy requirement, enzymatic function, muscular function, and cushioning, thermal insulation, maintaining the temperature of the body. These are the three important functions that are there in the body. So for this we have three major nutrients that we need to eat. We need to supply the body. So carbohydrates, proteins, and lipids, fats. All of these are interconvertible. Our body has mechanism to convert one into another form. That is there. But what we are supplying, these three are the major micronutrients, macronutrients. Carbohydrates are for what? Energy. Proteins are for muscle function and also to maintain enzymatic activity. Lipids are for cell membrane is also lipid, correct? For cellular integrity. If you starve yourself from eating lipids, if you have a completely fat-free diet, assuming that eating fat is what makes you fat, eating carbs is what makes you fat, because liver and every cell, every adipose tissue cell has the enzymatic machinery necessary to convert carbohydrates into fat. That is the evolutionary mechanism. If you eat too much of fat, the next day you will have diarrhea. That's all. It will not get absorbed and get stored. If you eat too much of processed carbohydrates, nothing will happen. Next day you will feel more hungry. Every bit of carbohydrate available in a digested form will get absorbed. But there is a threshold for the absorption of lipids. You cannot absorb everything. Okay, so carbohydrates, proteins, lipids. Carbohydrates and lipids are for energy. Proteins are for sustenance of the body functions. Minerals are for skeletal health, muscle function, blood functions, and nerve function. So minerals are very, very important, though in small quantities. Without calcium, bone becomes weak, muscle cannot function, neurons cannot function. So calcium is very important. Without sodium, neurons cannot function. Without potassium, heart will stop. Alright. So, like that. Without iron, your blood count will come down, hemoglobin level will come down, your circulation will be affected. Minerals we understood. What do you need vitamins for? What is the use of vitamins? Why do you need vitamins? Say vitamin deficiency disorders. If you don't take vitamin A, what will happen? You will develop night blindness, your skin will start becoming rough. Why? understood. But vitamins, why? All of you have your books. This is a room surrounded by a lot of trees, so if there is no light, will there be enough visibility for you to write? During the daytime you might still be able to manage, but in the evening sessions without light, none. So if to ensure that there is light in this room, who is required? Who is required to manage these lights? Who fixes the lights? Electrician? You need an electrician, correct? So whether there will be light in this room or not is dependent on the work of the electrician. Now let us say this electrician is dependent on his assistant. You go and tell you call the electrician, you tell the electrician is there, he is sitting right outside the door. There is no light in the room. You tell him, please come and fix the light. He says, my assistant is absent. So what, I will assist you, please come. No, I want that assistant only. Will the electrician be able to fix the light? Even if he is there and the light is there, everything is there. But electrician will work only if there is the assistant. The same assistant has to come and hold the chair, hold the ladder. Otherwise this fellow will not touch it. Alright. Some electricians, or let's say there are electricians for the fans. Let us assume all the fans should be working for the room to be ventilated. None of the fans are working. The electrician who is supposed to repair the fans is sitting outside. You go and tell him, please come and repair the fans. None of the fans are working, there is no ventilation. He will say, my assistant is not there. But I will work. There are 8 fans in this room. I can only fix 3. So will there be enough ventilation in the room? No. But there will still be ventilation. Correct? The electrician is the enzyme. Assistant is vitamins. Major role of vitamins is to function like coenzymes. Alright, the assistants for enzymes. So without, what is the deficiency in the eye for night blindness? Vitamin A or retinol is required for the production of what? What is the pigment in the eye? Rhodopsin. Rhodopsin is the pigment that is produced which will help you see in the dark. Without rhodopsin, you will not be able to see in the dark. Night blindness will happen. The enzyme which is supposed to produce rhodopsin is dependent on the coenzyme retinol, which is vitamin A. So even if there is the enzyme, vitamin A will not, if vitamin A is not there, this enzyme will not function. So vitamins are required for ensuring that the enzymes do their job properly. Alright. Let us say this fellow has to come. The assistant has also come. But the electrician says the electrician has to fix the lights. Electrician is also there. Assistant is also there. Electrician has a fractured arm. And another arm, he has 2 fingers broken. Now he has to fix it. Assistant is there, electrician is supposed to fix it. He will still try and fix it. But will he be able to do it efficiently? No. Why did the electrician get into this situation? Consider electrician to be like, okay, let's say this electrician is can be repaired in the hospital. There is a hospital in the building also. The doctor can fix this, but there are no materials to replace. His bone has to be replaced, broken bone has to be replaced. There is no bone available. These 2 fingers have to be recreated. There are no finger material available. Alright? So what do you need to make sure to ensure that the electrician is working properly? You have to ensure that the hospital has all the materials necessary to create these workers or repair these workers when they become weak and ill. What are those raw materials? Amino acids. This electrician is the enzyme. Enzymes are proteins. So if you don't supply enough proteins to the body, if you don't eat enough proteins, you will not have the amino acids necessary to make these enzymes. So the enzymes will either be absent if there is gross deficiency of amino acids, or the enzymes may not be fully functional. If the enzymes are absent, disease proportionate to that. If the enzymes are deficient, if the enzymes are not fully functional, discomfort or disease to that extent. Fully functional body requires energy in the form of carbohydrates and lipids, which generally everybody gets. Even somebody who is doing very menial jobs, who has very limited resources, they will also ensure energy is supplied. Proteins in the diet to give essential amino acids to the body. With using those, the body will manufacture the necessary enzymes which will keep the body healthy. Not that minerals in the right quantities so that all the essential functions, all the vital functions in the body are being carried out properly. Vitamins in proper proportions so that the enzymes can function efficiently. Production of enzymes. See, initially when we discussed the energy dynamics and the cell dynamics, I told you that this whole body, the functioning of this whole body is dependent on its enzymatic machinery. Yes or no? If all the enzymes that my body is supposed to be producing is produced in the right quantities, in the right way, my body will continue to function healthily. So to make sure that the enzymes are produced in the body, what I need to supply? Proteins. To make sure that these enzymes are working efficiently, what I need to supply? Vitamins. To make sure that the vital functions are happening properly, musculoskeletal functions. Musculoskeletal functions and the circulatory functions are happening properly, what we need to supply? Minerals. That's all. So maintenance of the body requires minerals, efficiency of the body requires vitamins, functioning of the body requires proteins, energy for the body requires carbohydrates and lipids. This takes care of our entire existence. Energy to function, carbohydrates and lipids, performance of all the functions, enzymes which are dependent on proteins, vital functioning, survival of the body depends on the vital functions, that requires minerals, efficiency of the enzymatic machinery requires vitamins. That's all. This is the same story for every organism. What organisms require which nutrient, which mineral, which vitamin, which chemicals in what proportions depends on its evolutionary history of survival. What it has adapted to survive, the chemical environment that it has created determines what it needs to take from the surroundings. For us, we have to take these 5. Alright? So, this is the story of biomolecules. Alright? I think almost the foundations for learning, 75% of the syllabus is there. Alright? Respiration and photosynthesis if I discuss, all foundations will be done. Then we can go into the concepts, discussing each concept as they come based on the paper discussion. Alright, wherever there is a need for elaboration on the fundamentals, I will go into that. Otherwise, tomorrow will be the last lecture based session. After that, tomorrow I will be completing most of the fundamental discussion. Friday and Saturday, I will complete the discussion on this paper. you will know how quickly we can learn with paper as the base. Alright, by tomorrow and today, by today and tomorrow, you should complete your analysis of biology portion of the previous paper. Alright, anybody already completed? 20-30 questions you are done of the 90? How many? 30. Good, no problem. Alright, any questions? Anything you need to ask? The first few weeks, keep pushing the physical boundaries. Push the physical boundary. You'll get tired, it's okay. Let us not be very slow in adaptation and take a longer time. Push the physical boundaries, you'll get tired, sleep during break time, get up, push again physically, keep doing that. Within a month or so, you'll completely align, and beyond that, body should not be your concern. Okay? If you are slow in adapting, if we take too many breaks, if you are too much worried about how our body is responding, all of that, it will take us longer for making our body shut up and listen. Alright? The body should shut up and listen. Close all the doors, it will listen. Okay, I understand it is not easy to sit for a 3-hour lecture. I can see on the faces, some of you are like pulling your head internally, pulling your hair internally in your mind. Some of you might be on the dais beating me up. Somebody is here already. Alright? It's okay. Alright. Continue with your analysis. Any questions, any doubts? Reach out.